Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your original guru of biochemistry. Well, today we are going to discuss again a very interesting and an important topic. We would be discussing about the metabolism of VLDL. Now what is this VLDL? How it is produced in the body and what happens to this VLDL actually. Now see, what VLDL stands for: very low density lipoproteins. Right, it stands for very low density lipoprotein. Now, first of all, we need to know that what is a lipoprotein. वैसे भी I have discussed this in my earlier previous videos also. Lipoproteins are actually the complex of lipid plus protein. What is the use of formation of these lipoproteins? Because lipids are non-polar, they are not soluble in water. So whenever they have to come into circulation, they will always require a companion, and that companion is nothing but a protein. And this protein part is a apoprotein. So this apoprotein, when it binds with this, it forms a lipoprotein. Okay, so simply we can say that now depending upon this, these lipoproteins are actually of four main types, right? So what are those lipoproteins? One is chylomicrons, VLDL, which we are going to discuss now, LDL and HDL. So there is one more. When VLDL is converted into LDL, LDL stands for low density lipoprotein, HDL stands for heavy density. So what do you mean by the term low, very low and high? So density depends upon the protein content. This is again very important to understand. Okay, so what happens is when this VLDL is converted into LDL, an intermediary density lipoprotein is produced, which is known as IDL. We can call it as a remnant VLDL also. So it's like one and the same thing. Right. Now, coming over to this, always remember this density is directly proportional to the protein content and inversely proportional to the lipid content. And this is how, just because of this particular reason, we can separate these lipoproteins by electrophoresis and by ultracentrifugation. Right? So the basic difference between very low density lipoproteins and low density lipoproteins is again the density or the protein content. Clear? So obviously if the protein content is more, the density is more. And if lipid content is more, the density has to be less. Clear? So if we talk about this VLDL is responsible for the transport of endogenous TGs. So these very low density lipoproteins are required for the endogenous TGs transport. Very important. Right. Now the question is, obviously from the liver to the extrahepatic tissues. And if we talk about chylomicrons, chylomicrons are responsible for the transport of exogenous TGs. So you have to understand the concept of your difference of endogenous and exogenous. Exo mane bahar, outside. So exogenous TGs are what? Which are coming from outside, or the dietary TGs. Okay. And if we talk about endogenous TGs are which are synthesized by the metabolism which is occurring inside the body. Okay. So VLDL plays a very, very important role in the transport of these endogenous TGs, and the body has to do what? It has to extract whatever is present in these TGs. And what is present in the TGs? The fatty acids and the glycerol. And other components are also there, like cholesterol is there, phospholipids are there are so many. Okay. So now what we are going to study is how this VLDL is converted into LDL, taking IDL as a intermediate or intermediary density lipoprotein. Clear? So now what happens, and always remember one thing, whenever we talk about the metabolism of lipoproteins, we have to consider the intestines, the liver, and mainly the adipose tissue, because adipose tissue is the main storage house of lipids. Okay. Now coming over to this, again I told you, since VLDL, they require a specific apoprotein, right? The moment they will come out from the liver, they will require a apoprotein because they cannot move on their own. So what are the apoproteins required for VLDL metabolism? So the first apoprotein which is required is Apo B-100. Along with Apo B-100, Apo C2 and Apo E. Right? Apo B-100 is specific for VLDL, and Apo B-100 is synthesized in the body by a process of RNA editing or processing. If we talk about the function of Apo C2 is an activator of LPL, which is lipoprotein lipase. And if we talk about Apo E is responsible for receptor mediated endocytosis. Right. So these are the three specific apoproteins for the metabolism of VLDL. Right. So first of all, I will just explain with the help of a diagram, then we will note down what are the important points in this. Now see, say this is the liver. Say this is the adipose tissue. Right. Now what happens is, or extraperipheral organs, we can say that. Now what happens, the moment this VLDL comes out, it is known as nascent VLDL. Okay, this is nascent VLDL, which is having endogenous TGs. And the apoprotein for it is Apo B-100. So this is Apo B-100. Now what happens is, it will start accumulating more and more TGs, and it will combine with two more apoproteins, Apo C2 and Apo E. And they will become mature VLDL. So obviously the size will little bit increase, and there will be now 3 apoproteins associated with it. So this is Apo B-100, Apo C2 and Apo E. Up now see what is the function of Apo C2. Apo C2 is activating LPL. Now what is LPL? LPL is lipoprotein lipase, which is already existing in the endothelium vessels like this. Lipoprotein lipase. So ApoC2 is going to activate it. The moment LPL is activated, it is going to extract whatever is there, because it is going to extract the TGs, because that is what is the funda of metabolism. Ki whatever lipoprotein is having in it, we have to extract that, so that the body, whenever the energy is required, this body will be, whatever is produced, that will produce energy when the body requires. Because TGs are what? Triglycerides formed of fatty acids and glycerol. And we all know that these fatty acids will undergo oxidation to give energy to the body during fasting or starvation. And whenever the body is in fat state, this fatty acid will combine with glycerol to form a fat or the TGs. And depending upon the proportion of this, they are designed like that. Okay. So LPL is going to take these TGs. And these TGs, obviously, they will be converted into fatty acid and glycerol. This we know. This fatty acid will enter in the adipose tissue, where it is going to combine with glycerol and it is going to form a TAG, and it will be stored in the adipose tissue. That is why we always say LPL is an anabolic enzyme. Just keep this thing in mind. Lipoprotein lipase is an anabolic enzyme which leads to the formation of the TGs. Ab now see, function kya hai? It is just extracting lipoprotein. Okay, just the protein part, dividing into this. Now coming over to this, now whatever is the leftover, that will be known as remnant VLDL. So the size has reduced, you can see that. So this is remnant. I will just write here mature VLDL. And this is remnant VLDL, which is commonly known as intermediary density lipoprotein. Now see, if we talk about the apoproteins here, apoproteins, Apo B-100 will always cling to it. Apo C is there, Apo E is there. Apo E. Okay, now the question is, now this Apo E will come into function. Because liver pe kya hai? The liver is having the antenna shaped molecules like this. And Apo E is responsible for receptor mediated endocytosis. So now see, whatever is left, whatever amount of TGs are there, this Apo E will be accepted here by receptor mediated endocytosis. Right, so this is one fate of this ideal, and second fate of ideal is that in the presence of hepatic or endothelial lipase, it is going to form a different structure, and that different structure is known as LDL. LDL, which has again Apo B-100 and Apo E. Right, this is the fate of this remnant VLDL. I'll just simplify it further. What I am trying to tell you here is that VLDL is responsible for the transport of endogenous TGs. Okay. Endogenous TGs are the ones which are produced in the body as a result of metabolism. So what will happen is, the moment this VLDL bearing endogenous TGs comes out from the liver, it will seek for a specific apoprotein, and that specific apoprotein is Apo B-100. Now this Apo B-100, when it combines with VLDL, it is known as nascent VLDL. This nascent VLDL now will take more and more of TGs, okay, because ultimate aim is to degrade them. So what will happen is Apo C2 and Apo E will come and help in this. Apo C2, when Apo C2 and Apo E combines, it becomes mature VLDL. And this mature VLDL is accepted by lipoprotein lipase. So this lipoprotein lipase will extract, right? And the leftover will be known as remnant VLDL. And obviously extraction will be there. Fatty acid will enter into the adipose tissue, forming triglycerides, and it will be stored as such, and glycerol will enter in the liver because glycerol cannot be metabolized in the adipose tissue, right? So now what will happen is this remnant VLDL or IDL has two fates. You can see that first fate is it will be recognized by receptor mediated endocytosis, or again TGs will be degraded. And second fate is endothelial lipase or hepatic lipase is there which will segregate this. So I can say that ये जो LDL बनेगा, it will have cholesterol or cholesterol esters with it. Okay, and if we talk about the structure of this, it is having TGs, it is having cholesterol, it is having cholesterol ester with it. Okay, but TGs are obviously more, right? So here also these TGs will be taken over and cholesterol will be carried over by this LDL, right? Now, what is the fate of this LDL now? Okay, so this LDL will undergo, again there are two fates, 70% of LDL, and you see Apo E is still sticking to it, right? So 70% will be again accepted by the liver. So I can say that again liver has antenna-shaped molecules which are going to accept this cholesterol. What about the remaining 30%? This 30% will move to the extrahepatic tissues. So it would be like in the heart and it would be in the adipose tissue. So I can say that out of means this LDL is having what? This LDL is having cholesterol and cholesterol esters. So what is happening is 70% still majority portion of cholesterol and cholesterol esters will be accepted by Apo E by the liver, and 30% will move to the heart and the adipose tissue because they have receptors for again LDL receptors, means to accept LDL. On the other hand, this LDL in the presence of free radicals, it will be accepted by the macrophages and it will lead to the formation of 𝗲𝗏𝗻𝗆. So it will lead to the formation of 𝗲𝗏𝗻s. Right? So this is the fate of VLDL. In some books you will find it as VLDN and LDL because LDL is actually formed from VLDL, taking IDL as an intermediate. And this is very important to understand because why the metabolism is important, because whenever we discuss about the different type of hyper and hypolipoproteinemias, these receptors are the ones which are either defective or there is a mutation. Okay, so remnant VLDL will be converted now to LDL and इसमें 70% will move to the liver and 30% will be accepted by the heart and the adipose tissue. And in the presence of free radicals, it will lead to the formation of 𝗲𝗏𝗻𝗆. And that is why we always say that this is a well-known fact, we all are familiar with this, that LDL is responsible for the transport of or it transports cholesterol from the liver to extrahepatic tissues. And that is why it is known as bad cholesterol, because it leads to the formation of atheromas, as you can see here, right? So this is the metabolism of VLDL. So let's quickly jot it down in the form of points. So I can simply say that first step is responsible for transport of, or you can say responsible for endogenous pathway. Right. Then what is a nascent VLDL? Nascent VLDL is only having Apo B-100 and it comes out from the liver. Right. Then what is the third point? Third point is formation of mature VLDL. And how this matured VLDL is produced? By Apo C2 and Apo E. They come from HDL. So still it is having more of the TGs. Then what happens is, since Apo C2 activates LPL, Apo C2 is activating LPL, so we can say that whatever TGs are there, automatically they will be extracted and the leftover will be known as IDL. So I can say that what is a remnant IDL? Remnant IDL is actually most of the TGs extracted, because LPL is an anabolic enzyme. Then comes, what is the fate of this remnant VLDL? So it has two fates, first and second. What is the first fate? Apo E, accepted by the liver. Right? And 2 is with the help of hepatic lipase, hepatic or endothelial lipase, it will be converted into LDL. And this LDL is having more of cholesterol and cholesterol esters. Right. And this LDL again has two different fate. I would say 70% I will write here and 30% I will write here. Okay, so 70% will be accepted by the liver and 30% will be accepted by LDL receptors in the peripheral organs. Right. So this is how VLDL is completely dismantled. This is again very, very important, right? So you have to remember these terminologies. एक तो you should know what is the difference between endogenous TGs and exogenous TGs? What is a nascent VLDL? What is a mature VLDL? What is a remnant VLDL? Remnant VLDL को we can call simply as IDL. And remnant VLDL का one path is it will be accepted by the liver, and whatever cholesterol is there, whatever cholesterol esters are there, they will separate out, they will pinch out, forming LDL. And this LDL again 70% will go to the liver and 30% will be accepted by, you can say, the extraperipheral organs, most predominantly the heart and the adipose tissue. Right. And this LDL free radical की presence में it is required for, in the presence of free radical, it is going to form atheromas formation. So this is how we explain the metabolism of VLDL, which is totally different from the metabolism of chylomicrons. Chylomicrons में the main thing which you had to remember was they are responsible for exogenous pathway. What is exogenous pathway? Exogenous pathway means the dietary TGs, the triglycerides which we are taking in the diet. Chylomicrons are responsible for that. Again, if we talk about a nascent chylomicron is, this is also very, very important. If we have to ask what is a nascent chylomicron? Right, nascent chylomicron is only having Apo B-100. And if we talk about nascent VLDL is having Apo B-100. Okay, and Apo B-100 and Apo B-100 are the only two apoproteins in the body which are synthesized by a process of RNA editing or processing, which we are going to discuss in the videos in molecular biology, when we will be studying, where we are going to study what is this RNA editing or processing, right? But you have to remember that there is a difference. Chylomicrons have more fat content in comparison to VLDL, right? And VLDL metabolism is associated with, obviously VLDL is there, IDL is there, and LDL is there. This is the broad beta fraction. VLDL is the pre-beta fraction and LDL is the beta fraction. So these are also known as the beta lipoproteins, and HDL is the alpha fraction. HDL ka metabolism is altogether different. So we can say that ki chylomicrons exogenous, VLDL endogenous. Okay, so whenever we discuss about the metabolism of VLDL, by default IDL and LDL will fit into that, right? So this is all about the VLDL metabolism, which is a very, very important pre-beta lipoprotein, which is responsible for the transport of endogenous TGs from the liver to the extraperipheral organs. So I hope this video is clear to all of you, and the most important among this is you should know how these lipoproteins are formed, in what form they are circulated, what are the specific apoproteins for them, and how they are actually degraded. And lipoprotein lipase is the most important enzyme, which I told you it is an anabolic enzyme. Just keep this thing in mind. Lipoprotein lipase is an anabolic enzyme and it is always activated by insulin. So insulin is going to promote this LPL enzyme. I'll write it here also. Insulin activates LPL. whereas insulin inhibits hormone sensitive lipase. On the other hand, glucagon always inhibits LPL. Why? Because LPL is anabolic in nature and HSL is catabolic. So, glucagon, epinephrine and thyroid hormones, they will be inhibiting LPL and they will be activating HSL. Very important. HSL we are going to study when we are going to study the beta oxidation or the oxidation of fatty acids, right? So this is all about the video on the metabolism of VLDL. I hope this metabolism is clear to all of you. Thank you so much.